Hey, so I want to make a video going over the membrane, the cell membrane or plasma membrane structure and function. So go ahead and pull out for me your notes on this chapter, and I recommend you have your notes in front of you. You highlight and follow along just like you would if you were sitting in the classroom. And so the membrane, or we call it plasma membrane or cell membrane, all of those things are interchangeable. It consists of phospholipid bilayer, or two layers. So you know what a phospholipid is. A phospholipid, remember, be able to draw this. I want you to be able to draw this. It's got a phosphate group head. It's got a saturated fat and an unsaturated fat tail. And remember that fat and water do not mix. Okay, and so remember that the tails are hydrophobic. The tails are hydrophobic. I mean, I always think about like cat feet. Cat don't like water. Okay, and the head is hydrophilic, and I think about it like a cat. Cats would drink water with their mouth. They're hydrophilic. Okay, the word polar means it has two different charges, and so this phosphate head has two different charges. So I remember that when I draw a pole through the head, it's polar, and the tails are, or the cat feet, the tails are non-polar. So here's what I want you to be able to, that's how you're going to remember it. The head is hydrophilic and polar. The tails are hydrophobic and non-polar. Okay, for the test, what I want you to be able to draw is a phosphate group, and this is a saturated fat and an unsaturated fat. Fatty acid tails. Remember, if it's saturated, it has hydrogens all over it. If it's unsaturated, it's missing a hydrogen. So it's missing one right there. It means it's unsaturated. Okay, so the cell membrane, that membrane all around every single cell has these phospholipids in two different layers going all around. If you look back at this first picture, you can see there's the heads, and there's the tail, there's the head, and there's the tail. Makes the cell membrane. So it's a phospholipid bilayer, contains a hydrophilic phosphate head region on the bilayer, hydrophobic tail. That's what we were talking about. Okay, there's also protein. So what is the plasma membrane made of? It's predominantly made of phospholipids and proteins. Proteins are scattered all throughout. There's integral proteins that extend throughout the plasma membrane, and then there's the peripheral ones that just stick on one side or the other. Okay, and let me show you this picture right here. You'll be able to see. So this one goes all the way through both. Okay, so it's INTE, goes all the way through. Peripheral is just on one side. You can see these words labeled here. Animal cells have an extracellular matrix containing protein fibers and large complex carbohydrates. These are used to support these are used for support and communication. So go back and make sure that you have highlighted, and I think, let's see. Oh no. Okay, make sure that you have highlighted that the phospholipid, the cell membrane is made of phospholipid bilayer and proteins, and then there's two types of proteins, so make sure you know all of this. Okay, it is liquid-like, it's moving, fluid mosaic model. At body temperature, the plasma membrane has the consistency of olive oil, so it's kind of wiggling all the time. Those phospholipids are loose and liquid-like wiggling. Cholesterol in the membrane stiffens. So cholesterol, we think about like animal fat or bacon fat, is high in cholesterol. Remember that unsaturated fatty acids are liquid at room temperature. And so that would be more like vegetable oil. Cholesterol stiffens. So that I always think about bacon grease and how it would harden as it cools off. Proteins are able to change location in the plasma membrane. So they're kind of wiggling all the time, moving throughout. Okay, there's also carbohydrates in this plasma membrane. Okay, carbohydrates. There are glycolipids, and that word, glycolipids, let me go back to my pen. Glyco is sugar, lipids is fat. So this is going to be a complex sugar attached to a phospholipid. If you look at the picture right here, you can see a glycolipid. Let's see if there's one labeled anywhere. So this is a phosphate. So a lipid attached to that would be a glycolipid. Now, there's one right there. So there's one labeled. That right there. Okay, there's glycoproteins. That's going to be a sugar attached to a protein. So glyco means sugar. Glycolipid is a carb attached to a lipid. Glycoprotein is a carb attached to a protein. Carbohydrate chains give animal cells a sugar coating called glycocalyx. This protects the cell, allows adhesion between two cells, and signal reception, cell recognition. So we have this sugar coating all around the cells, and it protects the cell. It allows the cell to stick to other cells. It allows the cells to communicate and to recognize each other. And right now we're just talking about cells, but it's so important when you do take, especially that second anatomy course, you're going to learn how, if you do take anatomy, you're going to learn how immune system cells communicate and they recognize each other. And so it's really cool. Carbohydrate chains serve as a fingerprint that allows the immune system to recognize self from non-self. And so make sure you know that the glycocalyx layer, so highlight this, the glycocalyx layer, oh no, I meant to highlight, there we go, allows for cell recognition and cell reception. That would be that glycocalyx layer. So a sugar coating allows for these things. Carbohydrates chains serve as fingerprints. So they, fingerprints are what make you unique, right? Everyone's fingerprints are different. And so that carbohydrate chain makes the cells unique and allows the immune system to say, okay, when your immune system cells bump into your cells, it recognizes your carbohydrates. And when it bumps into a bacteria, we'll say, that's causing an infection, it would know that it needs to attack that. It's a foreign invader. Okay, there's also channel proteins. It's just what it sounds. These are proteins that allow molecules to simply pass through that membrane. Hydrogen ions pass through channel proteins when the cells produce ATP, and so does water. Water passes through a special channel called an aquaporin. Carrier proteins combine with specific substances to allow passage across the membrane. An example is the sodium-potassium pump. Okay, so we have channel proteins. They do just what they say they do. They allow, channel proteins are going to allow some molecules to simply pass through. It's like a channel. Come on in, hydrogen ion, go out, hydrogen ion. Okay, carrier proteins combine with specific substances and allow, so they bind with something and they open and close. Sodium and potassium ions move through these during nerve conduction. Okay, cell recognition proteins, highlight all of this. Cell recognition proteins, or carbohydrate attached to proteins, glycoproteins, that help the body differentiate the body's own cells from invading cells. So the cell recognition proteins are so important for the immune system. That's what that's talking about right there, is differentiate body's own cells from invading cells. That's all about the immune system. And so our cells are so cool. They have these carbohydrate protein attachments that allow the immune system to recognize your cells from, a bacteria, a pathogen. Okay, there are receptor cells. These are shaped so that a specific molecule can bind to them. When the protein binds to the molecule, the protein changes shape. And so a receptor protein is going to receive a molecule, and when it does, it changes shape. And that way, so this cell membrane is so interesting because so many things happen in the cell membrane. It has channels, it receives molecules. An example, liver stores glucose, that's sugar. Liver stores glucose after a signal from insulin. And so insulin is the hormone in your blood that says lower blood sugar. So the sugar will go into the liver because insulin is like a key, it opens the door. Insulin lowers the blood sugar. The sugar goes into the liver. Remember, enzymes are proteins that speed up a reaction, right? They're catalysts. They speed up a reaction. So enzymatic proteins carry out metabolic reactions directly due to enzymes. Junction proteins are going to be involved in forming types of junctions between animal cells. This allows signaling molecules. So junction molecules, which I'll show you a picture, but junction molecules will allow, so like here's a cell and here's another cell. They can communicate through these little junctions, little tunnels where they can signal pass molecules from one to the next. So here's an example. Okay, so look back at your notes as we look at these pictures. I'm visual, so this will help. So the channel protein is going to allow molecules like this, and the example given in your notes was hydrogen ion. Okay, the carrier protein, look right here at this picture. You can see that molecule, like the sodium and potassium, will go through. It is very similar, but selectively interacts with a specific molecule. Okay, cell recognition. Okay, so that's the next one, glycoproteins that help the body differentiate your cells from like a bacterial cell. Okay, so this one has a marker on it. It has like a flag. It has a flag. It's holding up a glycoprotein flag, and it helps with the immune system, right? And that's what this is talking about, white blood cells and immunity. Okay, receptor protein. See how this molecule right here will fit right into the protein on the cell membrane. All of these are proteins on the cell membrane. Receptor proteins receive for communication. It's communication, right? These are enzymes. See that it's showing you this molecule goes in and it changes into that one. So the enzyme will speed up or catalyze the reaction. And then this is a cell, and this is another cell, and through this little tunnelway right there, they're communicating. Junction proteins join cells so that tissues can fulfill a function, like your skin tissues will stick together and communicate. Okay, so cell signaling. Listen, I tell everybody, whether you're an Alabama fan or an Auburn fan, your cells are Alabama fans, right? Roll, Tide, roll. If you can remember that, then you can get this right. It's reception, transduction, response. Okay, so this is a cell right here. Imagine this whole thing is a cell. It's just showing you a little piece of the cell. A signal molecule may be a hormone. So a signal molecule will bind to the receptor protein, and when it does, it's going to relay a message. And then the cell is going to respond. So reception is where the signal molecule binds to the receptor protein. Transduction is where the message, like the hormone, is changed into something the cell can understand. And then the cell will respond. Okay, so the cells of multicellular organisms will talk to one. This is how cells talk to one another. It's called cell signaling. The 3 steps of cell signaling are reception, transduction, response. An example, pancreas sends the signal molecule insulin through the blood to a liver cell. So just, for example purposes, this could be insulin right here. The signal molecule, maybe a hormone, the signal reception is where the signal molecule binds to the receptor protein. Well, then the receptor protein relays the message. That's transduction. It relays the message in and says, tell the liver cell to open up the channel and take in insulin. So that's an example, but it's so cool. Our cells don't talk through text message or Snapchat or whatever. Our cells talk through molecules. Our cells talk through this right here, this word signal molecules. So know those steps: reception, transduction, response. You may need to know that for the test. You may need to list and describe the 3 steps. I'll do it one more time. Reception is where the signal molecule binds to the receptor protein. Transduction is where the message is relayed inside the cell. And then 3, the response is where the cell responds to that message. Okay, the plasma membrane regulates the passage of molecules in and out of the cell. And so the whole cell, look at this, this is the cell right here, and this is the bilayer, phospholipid bilayer. And you can tell right here it's showing like water's going in and out, right? This right here says charged molecules, they're not allowed to go in. See how that arrow is? As a general rule, small uncharged molecules can flow freely. See right there, this says non-charged molecules go freely. But large molecules like macromolecules, so charged and large molecules cannot flow freely. So the plasma membrane regulates the passage of molecules in and out of the cell. The membrane is selectively permeable. Only some things can come in and out. Differentially permeable, meaning that water can go in, small, highlight this, small non-charged molecules can't pass freely, but the opposite, large and charged particles cannot go in. So CO2 and oxygen may pass through freely. Water is able to pass in and out of cells through a special thing called, highlight this for sure, aquaporons. Water passes through special channels called aquaporons. Okay, so pause the video for just a second and try to go back and just really look into that cell membrane again. Make sure that you understand the different types of proteins right here. Go back and look at this page right here and make sure that you understand the different types of proteins. The cell membrane is made up of 2 layers of phospholipids. There's carbohydrates attached to, if it's attached to a protein, it's a glycoprotein. If it's attached to a lipid, it's a glycolipid. And it allows small uncharged particles to come in and out. So pause the video and take a moment to look back over what we've talked about so far. Okay, so when we talk about, let's see, there we go. When we talk about transport, remember we said small uncharged molecules can move in and out of the cell membrane. Large charged particles cannot. So let's talk more in depth. We're going to kind of zone in about transport. Those 2 types, or there's a lot of types, but 2 big types of transport. We call it passive transport and active transport. So first we're going to talk about passive transport. Passive is easy. It doesn't require energy. Describes the movement of particles across the plasma membrane where, highlight, underline, circle, energy is not required. So passive transport does not require energy. This is easy. You got this. It's diffusion if it's any type of molecule that doesn't require energy. The movement of any molecule except water from areas of high concentration to low concentration. So if you look at the picture, it's showing, I believe it's showing oxygen. And the oxygen and the CO2 move from high concentration to low concentration without energy. Okay, let me give you an example. If I were to stand in front of the room, so just imagine with me, and I sprayed a whole bunch of perfume. Now imagine these little particles right here are the perfume in the air. Naturally, without energy, like no battery, no fan, whatever, the perfume molecules are going to spread throughout the room, right? And the perfume will not stay in that one cloud. It will spread out. That's diffusion. The movement of molecules from high concentration to low concentration, right? It's a high concentration of perfume at first. It's going to move to a low concentration without energy, okay? Osmosis is the same thing, except it's only water. Osmosis gets its own name. Diffusion of water across a differentially selective permeable membrane due to concentration differences. Water always moves from high to low. Okay, you can see in the picture, isn't that a nice little animation? You can see in the picture, there we go. You can see that the sugar is not moving, but the water moves from, so this is like, over here is like 100% water, and it's going to move over until it reaches equilibrium. So water from high to low, no energy. Osmotic pressure, the pressure that develops in a system due to osmosis. Okay, the solutes, think salt and sugar, suck. The solutes are going to pull the sugar in. Okay, an easy way to remember this, diffusion and osmosis both is, if you, this is just like a little easy way to remember it. If you were walking down a hill, so this is you walking down a hill, from high to low, this is high, this is low. If you're walking down from high to low, you don't require energy. Let's give you some arms. Energy is not required from high concentration to low concentration. See, high concentration to low, and that's osmosis and diffusion. It's high to low, no energy. Now, we talk about those solutions that you just saw, that water. If it's equal, it's isotonic. The solute, we're talking about the solute. That's the salt or sugar. The solute concentration and the water concentration are equal. Okay, hypotonic solution causes the cell to swell or even burst. Okay, hypo means low. Hypo refers to low or less solute. So you got to remember that all three of these things, iso is equal, hypo is low, and hyper is high. But what you have to remember is that we're talking about the solute concentration. Okay, salt or sugar is solute. It's either equal, the solute concentration is low, or the solute concentration is high. Okay, hypertonic causes the cell to shrink. Okay, let me show you. Let me draw this out for you. Okay, so here is kind of like what we were just looking at. That beaker of it was like a beaker of water, but they have a membrane in the middle of it. Okay, if there is, let's just say 50% salt on one side and only 10% salt on the other, the salt, we're talking about the solute concentration, the water is going to move towards the solute. The water is going to move towards the solute, and this side is going to swell. Think about a hippopotamus has a big bottom. Hypo, I know it doesn't fit just perfectly, but it's how I can remember it. Hypo, hippo, whatever. It's going to swell. So the hypotonic solution means that the solute concentration is less, so it's actually going to swell. Okay, another way to look at it, probably actually a better way, I just thought of, if you had like a beaker of water, okay, and in that beaker of water you had a cell. And we say that your cell is, 70% water, so it's 30% solute. And it's in a beaker of water, which is 100% water. 0% solute, so that should say 0%. 0% solute, 100% water. So which way is the water going to move? The water is always going to move towards the solute, and that's going to cause it to swell. The opposite could be said about low solute solution, causes the cell to shrink. And so if we put your cell in salt water, so this is a lot of salt, and this is your cell, then your cell is actually going to dehydrate. It refers to it is actually going to shrink. If your hyperactivity might make you lose weight. Just an example. Okay, so here's pictures. I know my pictures were better I know. Okay, iso, remember, is equal. No, water moves in and out at the same rate. Hypotonic solution, the water's going to move in, it's going to get big or burst. Hypertonic, the cell is going to shrink. Okay, there's also facilitated transport. Allows certain molecules to pass in and out of the membrane through carrier proteins along the concentration gradient. That means from high to low, with the gradient, moving with the perfume I sprayed at the front of the room. Except this time it's going through a protein facilitator. It's actually going through a protein. These channels are specific. The rate of facilitated transport depends on the number of carrier proteins for the substance. Okay, so take a minute. Let me, we don't need any of that. So take just a minute and let's look at this. So for passive transport, that's what we've been looking at. Okay, you had examples where osmosis, diffusion, and facilitated diffusion. Make sure you know the difference in all of those. Osmosis is the movement of water. Diffusion is the movement of all other molecules besides water through the membrane. These both go right through the membrane. And then facilitated diffusion goes through a protein channel. It actually goes through a protein channel, but is still facilitated diffusion through a protein channel. All three of these go from high to low, remember, up the hill to down the hill, high concentration to low concentration, and no energy is required. Okay, so let's go back to the notes now and jump into active transport. Active transport requires energy, and it's always going to be the opposite, from low to high. So highlight that. Requires carrier proteins, so it's not going to flow through the membrane like we talked about before. An example, highlight this, underline this. The sodium-potassium pump is an example of active transport, and it requires energy. It requires energy. So here's a picture. The sodium-potassium pump opens and closes, and it allows, in this picture, it's showing potassium entering the cell and the sodium exiting the cell. Large molecules enter and exit the cell by vesicle transport. Okay, if the large molecule is exiting, EXO, we call it exocytosis. If it's entering, ENDO, we call it endocytosis. Okay, so let me show you a picture. Let's see, let's go with my pen. Okay, so here's some food that the cell wants to bring in, if it's a bacteria or maybe some different proteins. So the cell is going to allow this food to come in. This is, oh, this is leaving. Oh, man. Okay, here we go. Here, start over. This wants to leave the cell. This is inside the cell, and it's going to form a vesicle. This vesicle is like a bubble around it. Find that word on your notes, vesicle, and it's going to push it out, and here it is exiting. So here's the particles in the vesicle. This is the vesicle right here. And it's going to push out, and now the particles are outside. Exocytosis is outside the cell. Okay, the opposite is endocytosis. Here's these particles, and they want to come in. And so your cell will take a piece of its cell membrane and pinch it off and make a vesicle, and that's bringing it all the way in or all the way out, and that requires energy. Exocytosis and endocytosis both require energy. Okay, if it's this is bringing it in. Phagocytosis is food. Penocytosis is liquid. Okay, so phagocytosis is like these to me look like solid particles, so they're coming in. But if it's liquid, then it's penocytosis. Not very scientific, but if you think like a pina colada is a drink, Pinot liquid, I don't know. That's how I remember it. So look at phagocytosis. White blood cells, like your immune system cells, will engulf and destroy the debris of lysosomes. They will bring it in and eat it. Pinocytosis, it says red blood cells, kidney cells, and plant root cells take in liquids this way. So P-I-N-E-I-N is to drink. The next one is receptor-mediated endocytosis. It's kind of like that signal transduction we talked about with the RTR. The receptor will receive the response and then open up a gate, allowing molecules to come in, like the response is to come in. Okay, so let's go back real quick to our Word document and look at active transport. Just, you know, if we were in the classroom, this is what I would write on the board. Kind of a P-O-R-T. It requires energy. Examples are going to be for active transport, the endocytosis and exocytosis. That's where these use a vesicle. Remember that bubble we looked at? Right? All of these active transports require energy. They use a vesicle. Types of endocytosis are, so kind of dig deeper into endocytosis. You've got phagocytosis and pinocytosis. And then another example is cell-mediated endocytosis. Another example of active transport, probably the most common example given, is the sodium, potassium pump. And all of these that I've listed here under active transport require energy. They're all moving from low to high. It requires energy when you walk up a hill. If you have to walk up a hill, that requires energy. Okay, so take a minute. I would pause the video right here and just like really look into the types of transport, active and passive transport. Now we're going to specifically talk about some cell structures that allow for communication. So animal cells have this extracellular matrix. Make that big again. There we go. Animal cells have this extracellular matrix. Like proteins, it is proteins and polysaccharides associated with the cell. And so it's like all around the cell, kind of like a skeleton, or extra cell. Extra means outside. Outside the cell, kind of holding it together like a skeleton though. Collagen, that's a structural protein. We even have collagen in our bodies. Elastin, flexible structural protein. And so you can see these proteins listed here. Of course, peptidoglycan, that doesn't say peptidoglycan, that says proteoglycan is an amino sugar that acts as a signal molecule. And so all of these are outside the cell in animal cells. These are protein structures. And if you'll look, right here it is, this picture will show you these. So this is the cell. And these are those protein structures like collagen and elastin, fibronectin, integrin, and proteoglycan. Okay, so this is pieces that help with the movement. So you can see tight junctions. Look at this. It's almost like little glue dots, right? Do you see that sticking two different cells together? The membranes of adjacent cells, like think of cells that are side by side that make up a muscle or a piece of tissue, typically join cells that form sheet-like layers. And so they're almost like glue dots that, you know what a glue dot is, that helps the cells stay together. Desmosomes are rivets or spot welds that are in skin cells holding them together. And these to me look more like Velcro. Okay, and then the last one is gap junctions. Tube, so that's right here. They're little tubes. Tube channels that hold cells together that let ions and nutrients move between the cells. And so on this slide, I want you to highlight for sure these tight junctions and gap junctions, right? Really all three of them, and desmosomes. So these are all three adhesion between two different cells, between adjacent cells. Remember, animal cells don't have a cell wall. So usually it's going to be the gap junctions, especially in animals that allow for that communication. Tube channels that hold cells together. This lets ions and nutrients move through, like heart and digestive smooth muscle cells. Okay, plant cells, well, they have this right here called a plasmodesmata. And that's, they have plasmodesmata, which allows water and small particles to be shared, right? Let me show you a picture. And so here it is. This is a plant cell. Let me switch to my pen. So this is a plant cell, and this is a plant cell, and they communicate between, it's like a hard plate. Right here, there's a little communication. You see that? They will have a plasmodesmata. It's allows a channel, allows for the plant cells. Plasmodesmata is found in plant cells. Let's see, right there it says plant cells. To share these particles. There's a primary cell wall. That's the cell wall. Do you remember what the cell wall of plants are made of? Hopefully we've covered it a couple of times in the class already. Cellulose is what's in plant cell walls. Okay, but there's primary cell wall. Some woody plants also have a secondary cell wall that contains more cellulose. There was that word right there. Okay, I hope that this was helpful. Please go back and study these notes and watch the video carefully. You can rewind or pay attention as you need to.